really no such thing as a direct path when you are thinking about navigating through college and graduate school. I was a first-generation college dropout. I dropped out of college, was working in the garden shop at Kmart. I was the best garden shop employee ever. And I sort of, a good friend of mine who's an organic chemist came in one day. He's like, hey, how's college? I'm like, oh, that stuff is not for me. He's like, what the hell is your problem, dude? And so he gave me a swift kick, and I got back and got myself sort of motivated. Then I had 2 kids when I was in graduate school. And so things just sort of, you find your way through this path. And, the most important thing is to keep it sort of realistic and to give yourself a little grace as you're going through this process and be patient with yourself because, it's not, we have this traditional idea of how this should be done, but almost no one does that. So I think that was a nice way you started that's sort of my story. And I am from Alabama, and so my folks were, college wasn't part of what they even knew about. And so my accent may come out up here, we'll see. Maybe it already is coming out, especially when I get sort of nervous. But I am an organismal biologist first and foremost, and I happen to work on ferns, and which makes my world at Colgate very convenient because Colgate's an undergrad institution where we have a fairly substantial research expectation, but we also have primarily teaching as our ultimate responsibility. And so all the stuff I'm going to talk about today really is work that I've dug into the archives to show the general talk, but also some of the more modern stuff is all done with some undergraduate students and some high school students. I've had a few postdocs in the lab over the years, but a lot of it really is with students. The workforce of my lab are undergraduates. And so I do want to talk about some of the work I've done. And I guess the approach I'm going to take today is to show you sort of the pathway that I've taken as an organismal biologist. I work with ecophysiological tools, but I'm not really wedded to anything. We're working on some stuff now with transcriptomics and fern gametophytes, which Dave Wickel informed me is not going to work, but the grant has been funded. Whatever tool we can use to throw at this group to understand the organism is what we do. And so I guess I'll first start out with why ferns, and I think in some ways it does sort of connect to what you were talking about, because here's this sort of cartoon tree of life. Ferns form this sister relationship with all seed plants, and so really everything we eat. So they have this interesting sort of story to tell us about the origin and the evolution of early land plants and what plants did as they moved out of these aquatic ancestral habitats onto dry land and how they still are connected to that environment. So we can learn something about ferns and the evolution of early plants by using ferns as a model system. Also, even here I would say there has never been and there never will be a group of plants as important as ferns. All of these rare earth elements out of coal and oil, they're all coming from sunlight that ferns fixed 300 some odd million years ago through the process of photosynthesis. And so they were around for a very long time, and we're digging them out of the earth and we're burning and releasing that sunshine, and we're going to be doing it for a long time. And so it's a really important sort of group for us economically and how we live our lives. And the field of sort of fern ecology and physiology, when I started 30 years ago now, I guess, good Lord, we knew very little about this aspect of ecology. They were really a group that we were studying evolution and polyploidy and multi-genome evolution in this group. But when we sort of sat down and started looking at ferns from a slightly different perspective, we learned some really interesting new things. Here's a paper by Tim Brodribb and Scott McAdams. Scott's a new faculty at Purdue now. Just looking at something so fundamental to the evolution of plants is the model function. They looked at ferns and said, hey, actually ferns have a really different way of opening and closing their stomata that doesn't follow this ABA hormonal mediated pathway. And when you can find something that kind of disrupts some fundamental understanding or some highly conservative trait, that's big news, kind of rewriting textbooks. And so just by a fairly simple experiment, finally looking at ferns in a different way, we've learned some really new, interesting things about the way plants work. So they're an interesting system, and they have a lot of lessons to teach us. Also, they're far from being of this sort of archaic importance back in the Carboniferous. If any of you have been out in the Northeast temperate forest, you've probably seen scenes like this. Ferns can form these monodominant stands and of dense, some monocultures or polycultures of species in the understory, and they act as sort of ecosystem engineers in our Northeast temperate forest. A lot of landowners where I work do a lot of stuff in the field, and they're always, all these ferns are horribly invasive. I said, no, these are native taxa. They're doing what they're supposed to be doing because they do suppress or at least control seedling germination, tree growth. they really do play overly important roles in our forest in the Northeast, which is obvious. But also a lot of our work has been over the years in looking at epiphytic ferns in the rainforest canopy. And they also play really important roles in the tops of these trees, and these emergent canopy trees. This picture was taken in Costa Rica a few years ago, where ferns make up almost always most of the species richness, sometimes a lot of the biomass in the rainforest canopy. And they're sort of the first plants, first interception point of nutrients and rainfall coming into these tropical forests. So they are ecologically important at multiple levels, and we don't necessarily think about them that way very often. I think now we do, but when I was your age, some of your ages, we didn't. And they do all of this with a double life, and that's been the focus of my career, really primarily on the gametophyte generation, but trying to understand how we approach the understanding of such an important group from the perspective of the organism in all of its splendor and all of its glory. How do we really understand this group, especially given that it has sort of this interesting sort of double life? And what I mean by that is, ferns and some lycophytes are this lineage of plants that have two independent phases. They have an independent sporophyte, the leaf that you recognize as a fern. If you walk through the forest, that's primarily what you're seeing. And unlike any other group, they also have this independent, free-living, multicellular, photosynthetic, in most taxa, gametophyte. And both of these things are out there on the landscape interacting with their environment. And if you look sort of a little more closely at these two phases, you see they have very different functional biology. And meaning that, here's a cross-section of a fern leaf, complete with cuticles, stomata, vascular tissue. It's like a normal-looking thing. If you look at this cross-section of a gametophyte, there's no cuticle, there are no stomata, there are no vascular tissues. It's a single cell layer thick for the most part of its body. And so selective pressures acting on these two different phases can be very different. In fact, they can be opposite, they can be opposing. So you have this complicated system that has a suite of, maybe potentially opposing selective pressures acting on it, but both of these phases have to function for the species to be successful, okay? And so the gametophytes is really what I focused on. I'm going to talk a little bit about that in a minute. But what's interesting over long evolutionary time frames, for whatever reason, and hopefully I'll convince you by the end of this that ferns are successful because of the gametophyte, not in spite of it, is that over evolutionary time, plants have moved in the direction of minimizing not the importance, but the sort of presence or dominance of that gametophyte phase, moving from things like mosses and hornworts, that group that no one really cares much about. The gametophyte is the dominant phase of the life cycle, and the sporophyte is dependent, parasitic, if you would, on that, to things like ferns that have independent gametophyte and sporophytes, to finally, the reduction of the male pollen grain to this, 3 or 4 celled, non-independent thing in seed plants. And so there's been general movement, sort of in reduction of the dominance of that phase. Which we could talk about if we wanted to later. But what's interesting about ferns, and which is the focus of this, is you may be sort of accustomed to thinking about the sporophyte leaf and what it looks like, and there's tremendous diversity in the form and function, I guess, but the form of fern leaves. If you go to the tropical rainforest, David and Peter, you've seen all this in the fern course I teach. There's an incredible diversity, everything from grass-like simple leaves all the way up to multi-divided compound leaves. There's a lot of diversity in morphology and form in the sporophyte generation. There's also an equal amount of diversity and form in the gametophyte generation, okay? So they really are, there's something going on there that is fairly unique at some very basic levels of morphology. And we knew about some of the variations in the gametophyte, but we've sort of, over the last 20 years, really tried to dig into this and think more about the importance and role of the morphology of the gametophyte generation. So, with all that said, how do you know this organism? Like anything else, if you were to give a talk and talk about, the biology of oaks or maples, and you never once mention the seedling, and ever considered a seedling and seedling biology, people would look at you like, you don't really understand this organism. And that's sort of where we were and not so much are anymore with ferns, because the gametophyte phase has been largely ignored. And so how do you get to know the organism? You really have to take a life cycle approach to understand how this thing works, because it's like a bryophyte and a vascular plant all combined into one. And so I want to take you through that process, of the way I've approached thinking about ferns from this organismal perspective through the evolution of epiphytism. We're doing a lot more work now in my lab on sperm physiology and polyploidy, but I wanted to dig back a little bit into the archives to kind of hopefully thread a story about how I think about and how my students think about this group and what we've been able to answer using the lens of epiphytism. So you may know epiphytes are those plants that grow on other plants, not necessarily parasitic. In fact, no ferns are parasitic, but they do grow disconnected from the forest floor. And epiphytism in the ferns has evolved many times, maybe 10 times, and then maybe more than that. So it's something that has evolved multiple times over the last, since the Cretaceous Tertiary boundary. And so obviously it's repeated evolution. And if you go up into the rainforest canopy, this sort of emergence or radiation into the canopy has happened really big. So here is a single branch of a Hieronima alcornioides, an emergent thing in the Euphorbiaceae in Costa Rica. I guess it's kind of hard to see the perspective, but this may be the size of a kitchen table or something like that. But if you look there are 9 different genera of ferns growing in this very small sort of liminal space. And so when I really sort of thinking about this group, I was like, how did these ferns get here? This is the most unfern-like habitat you might be able to imagine apart from the desert. It's a very bright, different kind of environment. So how did ferns get there? Because the ancestral condition was not there, was not in the rainforest canopy. And really to approach that, to understand that, we wanted to understand, how epiphytic ferns function relative to their terrestrial counterparts or sister taxa. And so that's sort of the work that has driven a basic question that has driven a lot of the work. And to sort of understand, again, I'll go back to the unfern-like habitat. The rainforest canopy is not quite a desert, I wouldn't say that, but it is, much brighter, much drier, very poor nutrients. There is soil up there, but there's very little soil, so it's a nutrient-starved environment. And yet you have all these ferns that you think of as being in a very different habitat that have irradiated and explosively radiated into this habitat. And so, here's some, this is just a simple, ancient paper we published a few years ago, many years ago now, looking at water potential over time at the first bifurcation. There's the sensor right there, that first bifurcation. It's a lot hotter, a lot brighter, a lot drier in the canopy. So it's a very different environment. And if you're thinking back to the 2 phases of the life cycle, you might be wondering just how they managed to do this with that independent gametophyte and independent sporophyte stage. So I'm going to take you on this journey. First, we're going to start out with this sort of haploid stage, okay? The gametophyte stage. And so what do we know, or what things might help us understand how they function in this environment? And one of the things I'll go back to is gametophyte morphology. If you just look at, I'm going to sort of break these into 2 sort of buckets, the epiphytic group and a terrestrial group, even though this phylogenetically is, mixed. There still are some functional groups that have emerged as these taxa have radiated into these different environments. If you step back and look at the morphology, there's a lot of variation in morphology, but there's a signal to that variation in morphology, being that, by and large, if you look at any biology textbook, this is what sort of you would see when they illustrate a fern gametophyte, this sort of cordiform, heart-shaped gametophyte. And this is, I like to tell my students, this is the sexual phase because it looks like a heart, and so, you would never have sex with someone you didn't love, and so, this is pretty typical, and this is what you see around here. If you look at terrestrial species, Dryopteris or other taxa here, that's what you would find. That's actually that's the minority morphology in ferns. Most ferns don't have that. I think almost now we're at the point where the majority of ferns might actually be epiphytic, but all of the epiphytic taxa have some variation on this form. Most always they're 3-dimensional, elongate. They really don't really ascribe to that sort of cordiform shape. So very early in my career, I was thinking, there must be some, given that we've had repeated evolution of epiphytism, and every time we see this evolution, we see a shift in gametophyte morphology, there must be some function. this morphology must play a role in this sort of mechanism play a role in a mechanistic role as plants these ferns have explored these different environments. And so does morphology have a function? So Riley mentioned Don Farrar was my master's advisor. Many years ago, he proposed a hypothesis, and I'll hopefully reconnect to why this is important in a couple slides down the line here, of why you might actually need some shape other than a cordiform shape. So here's an example of two gametophytes growing in a moss mat that you might find in an epiphytic situation, right? And these things still are connected to the aquatic system. They have motile swimming sperm. And if you had to swim a sperm through all this mess to get to this egg over here, the functional distance is really, really far. And so these sperm have a single mitochondria. They don't have a lot of energy. Some of them swim for hours. We didn't know how long they swim, but the idea was that it would take a long time to get there, and maybe that would be the sperm physiology itself might act as a selective pressure to change morphology, in that if you have a three-dimensional elongating indeterminate form, you might be able to kind of grow and get close enough that the sperm would be functionally close to each other, and you could make babies. And so that was sort of the leading sort of theory that Don had with Cynthia Dassler, who was a fantastic student in his lab many years ago. And so I went into this thinking that, and hopefully you'll see how my thoughts on this evolved over time. So I thought, okay, if Don is right, then there ought to be some fundamental differences at things like demography, how long these things grow, how long they live. When I started this work, if you asked anyone, how long do fern gametophytes live? Oh, 3 or 4 months. No idea, right? No one had really ever looked at something as fundamental as demography of this group. And so I set out to do this in a series of papers where I went out into the field, again in Costa Rica, found different taxa, marked the gametophytes in various creative ways, and I thought, good, this will be a good summer project. I'll go get these 3 months of data, everything will die, and I'll be good to go. It turns out that wasn't the case. I had to go back and back and back and back, because what I discovered very early on is that these things can actually be really long-lived. Okay, so here on this axis we have mean longevity. We have the 5 taxa here. The epiphytes are in green, terrestrial are in blue. These are epiphytic gametophytes, terrestrial gametophytes. And what you see is that there are some very interesting differences. The first thing we saw, people were like, oh my gosh, really? 2 years? Actually, I had to end this experiment this observational experiment, because they were still alive and I had to graduate. And I'll say that the last Campyloneurum brevifolium gametophyte died at 16 years old. And it died because the tree it was growing on fell. And so, first off, they're a lot longer lived than we thought. And in general, the epiphytes are longer lived than terrestrial. But if you also did look at the behavior of what happens, even though they may be living a long time, how many of them are actually living that long? And that's where things really divided. If you look at the percent of gametophytes that are still alive, you could either make a sporophyte or you could die. Most of the gametophytes of the epiphytic taxa were still alive. Very few of the terrestrial species were still alive, even though some of them could live for at least 2 years. Not many of them did. So there's a fundamental difference in something deeply basic, like demography, between these epiphytic and terrestrial taxa, really probably relying on the stability of the habitat in which they grow. So then that's the next sort of series of questions I had is like, well, wait a minute. If you're living out there for a couple years, you're living through wet and dry seasons, you must be experiencing quite a bit of stress. And, because ferns have this aquatic swimming sperm, everyone says, oh, the gametophyte is this weak link, and because they have swimming sperm, they can't grow in dry places. It's totally bogus. The sporophyte is the problem. But so I thought, there must be something going on with the stress physiology, especially related to water relations of these things that we thought were quite sensitive. We've published a lot of different papers on this. I just will pull an extract from a Frontiers paper where I sort of approach this from the perspective, if this were a leaf, how would it function? These things are a single cell layer thick, so there's no internal capacitance. They really can't store much water inside of those cells. But could it be that this three-dimensional shape might act as it does in some bryophytes to have this sort of exohydric external water holding capacity that might actually influence something about their ability to tolerate or live in these different environments? And so we did some very simple experiments looking at just drying rates, and you see that you dip these things in water, put them on a scale, and dry them out. You know, the Campylornum, which is an epiphytic, this is a terrestrial Pitiogramma gametophyte. They dry at different rates. No big surprise that if you're three-dimensional, you have all those interesting angles in your branches, you're going to dry more slowly. And then if you look at the ability then to remove this water, this is when I was my first experience as a molecular biologist. I used spin columns. So that was my experience with that. And putting them in there above those screens and spinning them at faster and faster speeds to see if I could remove this water. It's almost all external. And you know, these three-dimensional shapes really allow these species to hold on to that water. Okay, so it's sort of functioning as maybe in a morphological capacity, not only bringing things together, but maybe it's sort of functioning at the level of water relations. So then I've done a tremendous amount of work now really looking at water stress desiccation tolerance in this generation. And I'm going to show you just a small bit of those data. And here is the same two species. Well, this is a different microgram, two different species, same images, but the same idea. This is an experiment, some of the early work we did looking at desiccation. Taking gametophytes, growing them in the lab, cultivating them, hardening them, and then exposing them to different treatments, water potentials or humidity treatments. Here we have Microgramma reptans, which has a gametophyte very much like that. That is a twig epiphyte. Grows on these emergent canopy trees on the very twigs, about as hot and dry and horrible of a place you could be if you were a fern. And then here we have Diplasium subsylvaticum, which is an understory rheophyte growing in the really dark, wet, delicious, perfect fern habitat, as you might envision. And it turns out that if you dry these things out in these equilibrium chambers, and you let them recover, rehydrate them, 24, 48, 72 hours, you see pretty clearly that Microgramma reptans has an incredible ability to recover from desiccation, but this species does not. And relative humidity is a fairly useless term, but a relative humidity of 10% is something like minus 250 megapascals, really, really, really dry, deadly dry. And so these plants truly are sort of the gametophytes of some species, especially some of these epiphytes, have this incredible capacity to tolerate desiccation. They really enter this anhydrobiotic state, and they recover, which was a big shock to myself and to members of a lot of members of the fern community. And it's one of these things as a student, you're like, I've made my career, fantastic. I'm the first one to ever show this. I was so happy. And then I went to Peter Alpert at Massachusetts, an expert in desiccation tolerance. He said, I found this is great. He said, have you searched literature very well, Eddie? I said, of course I have. I'm a grad student. I've done my due diligence. That's my whole job as a grad student, to know thy literature. And he said, here, take this paper. It was a paper published in 1916 by Fernald Pickett, where he dried gametophytes out and they recovered. Not so sophisticated like this, but so I wasn't the first person to report this, but first person to use modern techniques. So you have to dig deep. It's a good lesson. So now we've done many more studies on this, and overwhelmingly, the epiphytic species have gametophytes that are significantly more desiccation, drought tolerant, temperature tolerant, light stress tolerant than these terrestrial species. Even if you control for phylogeny, look across the phylogeny, we see this repeated over and over and over again. So there's this sort of functional suite of sort of physiological responses that have evolved in concert with the movement into the rainforest canopy. That's not to say that if you're terrestrial, you don't have these skills. It certainly is the case that if you look across epiphytes and terrestrial species, they do generally have this pattern. But we just had a paper that came out, I guess, a couple weeks ago in New Phytologist with 2,000 gametophytes looking at this very similar thing in terrestrial wood ferns, looking at diploids versus allotetraploids, trying to elucidate whether or not the allotetraploid gametophyte phase had some sort of advantage over the diploid gametophytes. And what we found was that in general, the gametophytes of these terrestrial temperate ferns have some degrees of desiccation tolerance, not as much as we've seen in some of these epiphytic taxa, but that the allotetraploids are definitely more flexible in responding to drought and temperature stress in the level of the gametophyte. So it is widespread in the gametophyte generation in general, but there are some species, for example, Dropteris intermedia, which is up here, probably our most widespread common fern in the northeastern United States, has terrible gametophytes. they're so weak, they can tolerate almost no stress, and yet the sporophyte generation is fairly widespread. So more work to be done on that. Okay, so I think I'm hopefully telling the story about, maybe a surprising story, hopefully, that the gametophytes can be long-lived in general. They can be tough in general. And that those two sort of, the two groups of epiphytes and terrestrials can be quite different from each other in these character traits. So let me just pause and sort of take a slightly different tack now and talk about breeding systems, because this is really where ferns sort of shine, or really what makes them, I think, successful. And that is, if you take a look at, there's sort of three different ferns have a really flexible reproductive system. This is a classic example of one of the three sort of options of breeding systems. Here we have a sporophyte producing two different spores from the same sporophyte. Those spores grow into gametophytes. They fertilize each other. So this is what we call sporophytic selfing, which is the equivalent of selfing in a flower. You have two different, you have the anthers and the carpel. The gametophytes exchange gametes, and you have sort of selfing in that flower, sort of equivalent to that, right? So ferns can do that. And ferns can also outcross, not surprisingly. Two different sporophytes producing two different gametophytes with two different genotypes possibly coming together. They can outcross, obviously. But what makes ferns and some lycophytes wholly unique is the fact that they can do this, that they are homosporous plants. And this gets at sort of one fundamental aspect of botany and what really separates plants from animals and what makes plants superior to animals. And that is, in animals, gametes are made through the process of meiosis. In plants, that's not the case. Gametes are made through the process of mitosis. And so in this situation, you have this sort of gametophytic selfing moment where you can have a spore being made through meiosis. It grows through the process of mitosis to produce this multicellular gametophyte whose sperm and egg are created through mitosis. And so the sperm and egg, the gametes, are mitotic derivatives. And so for all intents and purposes, they're identical to each other. And where they cross, as I'm demonstrating in this example, what happens? Well, the emergent sporophyte is completely homozygous at every locus. And if it's successful, it's completely eliminated any recessive lethals that it has in its genome, because everything is expressed in the homozygous condition. And so you look at this and say, what on earth? This is a ridiculous evolutionary strategy. And I would agree with that. But it's a brilliant ecological strategy. If you can do this, and some ferns, a lot of ferns can do this, then you almost have sort of dispersal being equivalent to migration, right? Which is a really powerful tool, the fact that ferns can do this. And it's only the ferns and only the homosporous lycophytes that have this option. It's an incredible, again, evolutionarily not so great, but ecologically, and if you're in it for the long term, establish a population, move on, and you could be very successful. So we published a paper a few years back in New Phytologist where we did this huge survey, and we said the vast majority of ferns can actually do this. They can rely on this type of breeding system. Maybe they'll not do it very often, but they are all capable of doing this on occasion. But I don't want you to think that ferns they do have genetic diversity. They're not depauperate, but something is preventing this. There's cool stuff about the biology we could talk about of why how they avoid this, but they do rely on it in certain moments. Now, that's the sort of backdrop. Let's step back and look at why I'm talking about this. It turns out that this is another extract from that New Phyt paper, that there is a big difference in the types of ferns that can do this with the lens of epiphytic and terrestrial species. So we call this isolate potential. Basically, if you have high isolate potential, you can self, and if you don't, you can't. So if you take a look at the percent self sporophytes, epiphytes really tend not to do this. They tend to avoid this sort of selfing capacity, whereas the terrestrial species and these tropical things, this is not so. This is really well controlled for phylogeny, as a matter of fact. This is out of that huge paper. So there's something fundamental too at the level of breeding systems between these two groups of functional groups of ferns. And so mating systems vary, and it's fairly consistent. So the gametophytes between these two groups are different in almost every time we throw something at this group, looking at gametophytes, they're different. And these differences are repeated over and over again. So, the epiphytes have, the gametophytes are larger, complex morphology that allows them to be in disequilibrium. Some of the work we've done as well shows that if you dry more slowly, you're more likely to be desiccation tolerant. If you dry really fast, you're toast, even if you're one of these twig epiphytes. So having that gametophyte to regulate drying time seems to be functionally very important for these groups. More water stress tolerant, lower isolate potential. So there's a suite of sort of functional traits that are sort of we can map onto these different ecological forms, okay? So we've done a lot of work with gametophytes. That's just a bit of the work we've done. I do want to talk about the sporophyte generation and some of the stuff that we've done and continue to do to really begin to think about what's happening. I do think the gametophyte is the most important part of this, but sporophytes are also, they do play a role in the biology of ferns, unfortunately. But and so to talk about the sporophyte, we really have done most of my work, has been sort of in the realm of biophysics and biomechanics and hydraulics. And I started this work back when I was doing my postdoc with Missy Holbrook at Harvard. And I wanted to cover, before I get started, two sort of concepts in thinking about hydraulics. Because the hydraulic literature is rife with confusion and infighting and methodology, which is just crazy. And so there are two basic concepts I think that are important in thinking about how water flows and moves through plants. And that is the concept of conductivity, how fast water can flow, and then how safe that water can flow. And so the conductivity, fairly straightforward. Here is a cartoon. And so we model flow through, through this plumbing or vascular system of plants, just like your plumber. I guarantee you every plumber in the country knows this. They don't know this equation, but they definitely understand this. You have to understand this to understand water flow. So the hydraulic conductivity, the rate at which water flows through the leaf, is related to things like productivity and photosynthetic rate in general. Faster flow means higher rates, you do better, you grow faster in general. And it's really controlled by a few fairly simple parameters. There's something about what you're moving through, the viscosity, if it's glue or water or syrup, how far the path is. If you've got to move it a centimeter or a kilometer, that's going to slow down how fast the resistance that builds up over length. And then it's also, how hard you push. The pressure potential, if you push really hard, it's going to move faster, all things being equal. But really, more importantly, is something about the geometry of the system in which you're pushing it through. So this Hagen-Poiseuille equation has that radius raised to the fourth power. So a very small shift in the diameter of the radius of a pipe results in a fourth power increase in flow, right? Fantastic. So now we understand how this works. All I have to do is just increase my conduits. The bigger my conduits are, I can increase my flow. It takes very minimal increase to have a fourth power increase in flow. And I can prove it. Look, see how much faster this guy's doing? So that is sort of the basic concept of efficiency, of conductivity and efficiency. Now, the caveat to that is not quite that simple, as nothing ever is, in that there's this concept of safety. Because if you think about the way plants move water, it's not being pushed out, there's no pump, it's being pulled physically through the stomata with the different water potential inside and outside of the leaf. The stomata open, that water is being pulled out of the leaf itself. And what happens if the stomata are open, water's moving, the soil begins to dry up or whatever happens, it starts pull that negative pressure, or that vacuum in that solution can become so great that the gases that are dissolved in that solution can form a bubble, an embolism, or cavitation. And that can occlude that conduit. And I have, I don't usually walk around with a syringe, but it's a really great thing if you do teach this. I think I'd probably degas this. You can demonstrate this really great for your students. If you just take a regular syringe, and you up in the front will have a good vision. If I just pull on this syringe, I'm putting see the bubbles forming in there? You can see that I'm doing the same thing. I'm boiling this water because see what's happening? So I'm cavitating this water, and I can do this enough that I can degas this, but there's all these dissolved nitrogen and things in this, and so that's what happens in plants. Because they're pulling so hard, or freeze, thaw, they can induce cavitation. The problem with that is that it can be bad. It can be deadly. It can occlude the entire vascular system. It is a redundant system, so they have these tracheid pits that water can go around, but if the plants don't shut this down, that embolism can spread, and it can be bad. It's a good reason why trees regrow their xylem every year, especially in these cold climates because it's all cavitated and basically toast. Can they recover? Yes, there's some evidence they can recover. It's not very clear what the mechanisms are, but they can recover. So, the concept of safety can be sort of evaluated or tested by creating these PLC, or percent loss of conductivity curves, where you dry things out and you measure their conductivity over time, over water potential or whatever you're doing, and you can sort of make various calculations there, P50 or P25, or the percentage of when they lose 50% or 25% of their original conductivity. And that can be a useful tool to compare across taxa. It looks easy. These are really hard data to gather. So, two concepts: anatomy, morphology, and then the safety. So let's step back now with that sort of introductory lecture and look at what we know about the anatomy of these epiphytes versus terrestrial, the xylem system in sporophytes now. And so here we have very simple trachea diameters across these different groups of epiphytes, some other things here, and terrestrial species. And you see the epiphytes have a very small diameter conduit relative to the terrestrial species. So you should be thinking, okay, well, they're not going to be moving water very fast if that's the case. But it also turns out the secondary concept is that it's much harder to pull a bubble in a very small diameter conduit than it is a large diameter conduit. The larger the conduit is, the more likely it is to cavitate in general. If there's any hydraulics person in here, I'm going to be ridiculed, I'm going to be destroyed. But in general, the larger the diameter of the conduit, the easier it is to cavitate. So if you're growing in a drier environment, you're going to have a trade-off between safety and efficiency, right? And that's what we see, we see that, at least the beginnings of that, in the trachea diameters. They're small, but, so maybe they're safer relative to the terrestrial species, okay? And what's interesting, too, about this is that this is sort of a small data set, but here's the conductivity itself, and here's mean hydraulic conduit diameter. Epiphytes are in open circles, terrestrials are in the gray circles. Over a given sort of range of hydraulic conduit diameters, the epiphytic conductivity doesn't vary much at all. There's a lot more variation in these terrestrial species in this window, and certainly beyond that window. So even if you do have variation in conduit diameter, the epiphytes can explore that in terms of increasing their hydraulics. And a lot of that probably comes down to this. What's really happening in these in-pit membranes, the in-wall membranes, which I won't talk about, but there's other stuff going on behind this. But if you then look at the safety, it turns out that we have many different groups here, but here are the epiphytes, and here are the open field sort of xeric terrestrial species. In general, they're not significantly different, but they do have higher P50 values, meaning they're safer. They're much more resistant to cavitation in general than are many of these terrestrial species, especially the species in mesic habitats like that Diplasium subsylvaticum, who I talked about, the gametophyte. So the sporophytes are very different between these two groups in terms of their anatomy and the way that anatomy functions in those different environments, and it's probably related to where and how they grow. And it does definitely control, I think, aspects of the morphology. If you go to these tropical rainforest canopies and look at ferns, they look different. Mostly the epiphytes are simple leaf, but they really have very, very short petioles or stipes, whereas a terrestrial taxa can have very long stipes. And if you go back to that equation, you look at that equation, the Hagen-Bacillus divided by something times the length, that length is a parameter that if that's one way you might be able to deal with your small diameters, is to shorten the distance in which you're moving this water. So hydraulics is really driving, I think, a lot of the morphological features that we see in the sporophyte generation. And there's an interesting phylogenetic pattern here. You guys know this. I should have asked you this question. Shoot, I should have asked you your Eupolypods 1 and 2 morphology. But there's an interesting sort of phylogenetic pattern between this sort of arrangement of vascular bundles in the stipes of ferns. We have a really exciting paper that's out for review in some really important journal that actually went out for review that hopefully we'll hear about in a couple days, about the actual function of this sort of band of tissue around these vascular systems that may be a novel way that ferns avoid, or at least recover from these moments of cavitation. So, sporophytes, same sort of thing. They're different. Small conduits, conductivity, but more, right? So I didn't talk about this, but they are more stress tolerant in general. So it's sort of setting up this, not quite a straw man, but at least a functional way to approach this by thinking about the two different ways on these extremes that you can be ferns, independent of the evolutionary process. This has sort of repeated itself over and over. This sort of first strategy where you are, strategy one, you grow, the cordiform gametophyte has to move fast because these terrestrial things, they don't grow in the leaf litter, they're growing on soil, the soil's gonna get washed away, you have to move fast. Sort of an R-selected morphology. Simple morphology, it means you're stress sensitive, but you might have greater selfing, but you can produce a sporophyte. But that sporophyte's gonna have larger conduits, it needs to grow fast, increased conductivity, decreased safety, but at the end you have a happy fern. It works. It works just like any other fern, just adapted and acclimated for its environment. You have the second strategy, which is more of this sort of xerophytic or epiphytic fern strategy, where you have a slow-growing gametophyte that reveals itself in the more complex morphology that can do two things possibly: increased water holding capacity, increased outcrossing. That slows your drying. The drying makes you more stress tolerant. Probably feeds into longer time in residence, so you can outcross more. That leads to a nice happy sporophyte with smaller conduits that has decreased conductivity, but it's safer, but you're happy, right? It works. This little guy is now 18 years old, so it's a long time. But he's happy because he has ice cream there. And so I think ultimately what I want to say here is that, I really firmly believe in this organismal approach. It's so easy and it's so exciting to be in the lab and be lost in the pipette and the primers and the machine that's measuring, and never step back and look at what we're looking at, the organism. And it's so important because none of us are here because we woke up as 7-year-olds and said, oh, I love DNA. Oh my God, DNA is so cool. It's so important, but something about that organism, whether it was a polar bear, something attracted us to it, probably was something that we could see. Right? And so I challenge you, I guess, to reinvigorate that organismal side of you when you think about the organisms you're working with, because it is such an important way to think about these plants, and we sort of lose the ology and what we do sometimes without looking at these organisms. So here's a nice way to close. We all love ferns. There's our heart-shaped ferns, and I'm a gametophyte, and I'm happy to take any questions should you have them. And I did bring some gametophytes if you have never seen them. I mean, you all know Fei Wei, so you've probably seen some of these things. So I'm not going to pass them around now, but if you want to see them, I have them. Questions? Yep. I think we need the mic because there's some folks online.
So I just had a question regarding that, kind of just an idea of how do the actual sporophytes get on top of a tree in the epiphytic fashion? How did they get there? Did they? I'm not sure if you can.
You mean from over evolutionary time?
Well, you showed the image with the 10 on the canopy of a tree, and so I'm just curious how they actually end up there.
They do produce spores. The spores are blown about by the wind, and they just land in these habitats, form gametophytes. The gametophytes are successful. They sort of just like a seed or spore dispersal, the same way other epiphytes get there. Okay. Over evolutionary time frame, the stepping stones of how, if you look back at these different moments of radiation to the canopy, it happened probably one of two ways, either in really homogeneous cloud forests where the soil and the trees weren't that different from each other, or through an intermediate step where there's this lineage of ferns that are sort of hemiepiphytes that live in both conditions, possibly a sort of stepping stone over gradual time radiating and evolving into that habitat. So evolutionary answer and an ecological answer, I guess.
Thank you.
So this kind of
yeah, there you are. Sorry.
This kind of intragametophytic selfing that you were talking about, and how it was lowered in the epiphyte morphology, I suppose. Do you think that has to do with the fact that your idea that there was more epiphytic ferns than there were terrestrial ferns, and that they have more adaptive radiation and larger blooms in general?
I think what it is, if you have time, then you can, I think from the gametophyte's perspective, there's this sort of one gene, one protein idea from 1 million years ago. It's one gametophyte, one sporophyte idea as well. That's true if you're terrestrial. It's not true if you're epiphytic. An epiphytic gametophyte can be big and produce many sporophytes over space and time, but the terrestrial thing is going to die. It's going to die either from the environment, or it's going to die when the sporophyte emerges from it. If you have time to relax and grow, then maybe you have mechanisms, whatever those mechanisms that we don't know what they are, that prevent self-fertilization. You can just hang out there and wait. And so there's something that's happening at the level of maybe the egg-sperm boundary that prevents this from happening, because they can. Because intragametophytic selfing is great, but it's not so great. Because you don't want to rely on that too often. So I think it's just a matter of their sort of functional ecology. They're out there for long periods of time, so they can wait for this. One thing that's fascinating is some brilliant BTI person that thinks about this. If you grow some of these gametophytes you can grow them for years in the lab, and they will sit there and you can water them, and they will never make a sporophyte. And then one day they're like, oh, ready. And whatever those limitations are, the genes or whatever, they're turned off, and they say, okay, I've done this long enough. Now I'm going to make a sporophyte. So those isolating mechanisms break down somehow. No one's ever looked at that stuff. So I don't know if that answers your question. I hope it does.
Yes, thank you.
I had a question about the longevity of the epiphytic gametophytes. Maybe this is a sporophyte-centric question, but I'm just curious, what the point is. If they're living for 16 years, are they dormant, just kind of waiting to happen to get fertilized? Are they growing? Do they have their own processes happening?
Yeah, I mean, that's a very, very good question, actually. And it's, they're just like anything else. I mean, if you go around here and you walk in the forest, your gametophytes are probably the size of your pinky nail, which I have in this thing. But some of these in the tropics can be dinner plate. They're huge clones. They just grow and grow and grow and fragment, and some of them have asexual propagules, and so they can get really quite large. And so they do continue to grow, and just like any other sort of plant, leaf would grow or clone would grow. And in fact, I think the sporophyte is such a drain on the system that some ferns have given it up completely. And there are many taxa that rely on gametophytes only and never make sporophytes in the field. We're finding that more and more common now in our floras, especially in subtropical floras. So, they're kind of going back to the good old days of, bryophytes and, because you have a system that is desiccation tolerant, pretty tough. Why would you give that up? So,
Thanks.
Yep, right here. Oh, you have a mic.
I'm going back to gametophytes.
Yeah, good.
So you showed the study that with the allopolyploids.
Yeah.
And so I'm wondering, is it the hybrid nature of it, or is it the ploidy? In other words, is there a way to make autopolyploids of the things that you're looking at to check?
Probably there are, but these are, that's a good question. And that's the next thing I was talking about, that we've done all this work with the physiology. Now we have all the RNA extracted. We're doing the transcriptomics now to see the idea. Is it because you have 4 copies of those genes? Or, these things often when they're the allopolyploids behave more like one of the parents than the other. So are the genes of that parent the ones that are controlling the desiccation or whatever? So we're looking at some of that, but we haven't, it's funny, Amanda Gruz, who Amanda Gruz gave a talk just a couple of weeks ago at IBC in Madrid, and I talked to her. She works with a lot of autopolyploid desert ferns, and that idea came up. Like, can we do this with the autopolyploid and the allopolyploids of the same species, so to speak, to test some of those questions? I don't know the answer to that question. I don't, whether it's homologous recombination or if it's some other sort of vigor that's arising from the hybrid situation. I don't know. It's a really good question, though. Yep. Alexander.
Thank you for remembering my name. You went into good detail about how ferns, terrestrial ferns are ecological engineers. Like they regulate, or I guess kind of govern what saplings can root, and they have cover much of the ground floor. But what kind of, and you briefly mentioned that a lot of water first goes through the epiphytic ferns, but could you go into more detail about what exactly they're doing, what kind of, how they're contributing to nutrient flows to the water cycle? I guess, could you elaborate more on their role?
Yeah, so I guess a couple of ways I would respond to that question about the sort of ecological importance of epiphytic ferns. If you go to the paleotropics, it's not unusual to find a massive bird's nest fern that might weigh 1,000 pounds, and it's a giant sponge full of insects, and those things will gather and hold water, nutrients. Because the soils are so poor in many of these tropical forests, they do rely a lot of on this atmospheric deposition coming in. So it goes to the rainforest canopy. It's gathered up by those epiphytes, and through stem flow or flow through, it goes down through to the forest floor. So whatever's up here is going to have first dibs and deplete or either enrich that as it moves through there. And so those epiphytic ferns can be that sort of sponge or filter, epiphytic taxa in general. Ferns play a role when they're in great numbers. But they do contribute. It's not uncommon to go up. My wife is a canopy biologist. She taught me how to climb. She likes to point that out all the time, but she worked on canopy soils for her dissertation, and you can have serious soils developing in these canopy systems of many centimeters deep. It's all organic, and there's very little inorganic matter there, but there's soil there, and it's all the epiphytic plants and ferns are creating that soil. They're sort of engineering that environment in the canopy as they're engineering some of the environment on the forest floor as well.
Wow, that's wild.
See, now why are you doing hornworts? You need greater purpose in your life.
Hey, it was only 10 weeks.
Good. You're not contaminated yet. You're not done. We're not lost yet.
From the ecology to the evolutionary, you brought it up early, and then is there the connection between the strategy 1 and strategy 2, sort of like how does one think over, I guess it's different terrains too, so
yeah.
Is there a simple or general story, maybe an energetic story around in there?
You mean in terms of the different functional strategies?
Yeah.
That's a really great question. It's shocking how fast this happens and how quickly it happens. I just was at the International Botanical Congress in Madrid, and there's a colleague from Taiwan, Cheng-Wei Cheng, who's working on this group of ferns that are very closely related to each other, and it happens to have 2 terrestrial members, and the rest of these things are epiphytic, and their terrestrial ones are mixed within that phylogeny, and both of those things have these cordiform gametophytes, and all the others have these 3. So I think it happens, it's very labile, I think it happens very quickly. Is it more energy costly, the energetics? I don't know. I mean, you're talking about sort of
something driving the evolution. What are those factors?
I think it's water. I mean, my master's advisor says no. The reproduction is going to be much stronger selective pressure to that. But I think it really is water at that level. These things are they're really in a constant state of equilibrium with their environment, and that's temperature, yes, but I think the water relations really is what is driving this.
Okay.
I think.
All right.
Yeah.
Thank you.
Yeah, we have a question here. Over here, okay.
This isn't related to the talk. I just wanted to ask how doing research at like a primarily undergraduate institute is different than something like working at like an R1, and how does that affect your research?
It's fantastic in some ways. I don't have to worry about these students' jobs or lives, so it's a little easier because that's your job. I'll send them to you all, and you all can worry about them. Plants are a hard sell. People want to work on polar bears, and I understand that. And so, so for that reason, I have a very open policy. Like, you want to come in my lab, work in my lab, fine. You have to work on ferns or something remote. I have a student working on bees now. That's a long story. But I'm really open-minded to what they want to do because I'm tenured now, and I'm allowed to be way more flexible, and I'm not dependent on grant money. And that's helpful to allow me to let the students sort of choose their path, and come in with ideas, and usually sit down with them and design something, even if it's outside of my area. So that's, I think, a little bit different. Fortunate a place like Colgate has a lot of resources. Not all PUIs have that, and so we have tremendous support for the research that we do, and undergraduate research is really fundamental. I was an administrator for 6 years, and I ran the summer research program. We spent almost $2 million in summer research funds for students at Colgate, and so we have those incredible resources. That's a huge relief when you are at a well-resourced instance, but not all schools are like that. But I really intentionally sort of talked about some of this older stuff that I've done, because there's so much good science you can do with a piece of string and a ruler. You don't have to have $1 million sequencer or a $80,000 light core. There's so many important and really interesting questions you can ask with very few resources, and things that are less intimidating to some undergrads that may not know how to use or be intimidated. You walk around with a light core that's $80,000. That scares the hell out of them. Like, if it breaks, it breaks. But so I'm not sure if I'm answering your question directly, but
you're doing. Thank you so much. That's really awesome that you're that.
Yeah. So, yep. In the middle. It's just there are people online, so that's fine.
Very edible ferns, maybe it's the lunchtime, so I'm thinking about that thing, but in general. Another question that got me thinking, like, since you said that the gametophytes are long-lived, so do they have some genes that are common, like similar to angiosperms or something? So I was just thinking about those things.
So are there edible ferns? Yes. I'm offended that you would ask that question because it's cannibalism. No. There are, so the ostrich fern that is commonly grown, people eat. If you go here in the spring and you buy fiddleheads, that's almost all, it better be all be ostrich fern. Most of them are very toxic. Pteridium is eaten in Asian cultures. So there are some edible ferns, but it's not a primary source of but there are some edible ferns. And genes, who knows? I mean, it would be fantastic. We don't know anything about longevity and the genetic basis for longevity in these gametophytes. I have no idea. I don't know maybe there's something known in mosses and bryophytes. I don't know, but it's a really interesting question for sure. Yeah. Sorry, I don't have an answer.
Thank you for giving the talk today. I thought it was really interesting. I was wondering if you studied anything related to the conservation aspect of ferns, or if this research has impacts on that.
We do. I mean, we just a few years ago worked on the American hartstongue fern, which is an endangered species that happens to have been first discovered up in New York near Syracuse at Clark Reservation, which you should all visit if you haven't been there. A lot of ferns. And a student with undergrad thesis that worked on this, really trying to explore the species is endangered and the numbers were plummeting, trying to understand if there was a gametophyte mechanism, if the gametophytes played any role in that. He did a brilliant, this is Wes Testo's work, y'all know Wes. He did a brilliant thesis, and he was able to show that in spite of all of my years of saying gametophytes are great, they're tough, the gametophytes are terrible in this species. And they're outcompeted, they're stress sensitive, they can't handle temperature, they can't handle water, drought. So they're really at the edge of their physiological tolerance zone, and I think that's probably a mechanism driving their extinction. But that's probably about as most applied thing I've ever done in my career, and so I guess that's sort of conservation biology. We work with the U.S. Fish and Wildlife Service, and I could talk all day about the species, but I won't do that. So that's the closest I've really come to conservation.
Thank you.
Earth elements.
Not about rare earths, but excellent talk. Thank you so much.
Thank you.
This brings to mind the question of climate change and your research over time and people before you. Have you seen any effects on climate change, especially in, any of the types of ferns that you've been studying?
Oh yeah, for sure. It's interesting because, I've been working in Costa Rica for many years, now going to Colombia. Just came back from Tatama. For many, many years, and even at, for example, the La Selva Biological Station, the species that I was seeing back in 1998 are really no longer there. Many of them have been lost, and many of them have reduced in numbers. So I've definitely observed that in my career. And part of this polyploidy thing, the journal, the paper we just published, had that sort of angle, like, are polyploids are going to be a rescue mechanism for ferns? Are they more stress tolerant? Or is environmental stress sort of selecting for higher ploidy levels in ferns? And maybe the answer might be yes. So definitely, there's a lot of concerns about this. I had a big project years ago on Polystichum unitum in California, because that species plays a really important role in the redwood forest. If you watch E.T., you're too young for that, but you see all those ferns in the understory. That's Polystichum unitum, and it's just a dense carpet, and it's disappearing because it's so dry in the southern part of its range. And we did this nice ecotone across California, and these species just have very little plasticity. They're just all pretty sensitive, and so as that drought moves northward in California, it's going to probably impact some of those redwoods even, or at least the way those forests behave. So I'm not sure if that answers your question directly, but I think they do play, that you can't perturb or lose a group like this and not have some ecosystem level processes, because they're so important.